IGCSE and O-Level Smart Biology M. Rashid Chatha 030046104457. 5. Enzymes. Catalysts are substances that increase the rate of chemical reactions and are not changed by the reaction. Enzymes are proteins that function as biological catalysts. Enzymes are important in all organisms because they maintain a reaction speed needed to sustain life. The substance on which an enzyme works is called the substrate. After the reaction, a product is formed. An enzyme and its substrate have complementary shapes. Enzymes are affected by pH and temperature and are usually denatured above 50 degrees Celsius. Enzymes tend to be very specific in the reactions they catalyze due to the complementary shape of the enzyme and its substrate. When an enzyme catalyzes a reaction, it forms a temporary enzyme-substrate complex before the product is released. Changes in temperature affect the kinetic energy of enzyme molecules and their shape. Enzymes can be denatured by changes in temperature and pH. Changes in pH affect the shape of enzyme molecules. Enzymes follow lock and key hypothesis. The enzymes a cell makes are determined by its genes, protein synthesis. Enzymes control the metabolic reactions taking place in cells. Enzymes have an active site where the substrate binds to the enzyme. When the substrate fits into the active site, the catalyzed reaction takes place. This is called the lock and key hypothesis. The activity of an enzyme is affected by temperature. Up to an optimum temperature, a rise in temperature causes an increase in the rate of reaction. Above the optimum temperature, a further rise causes decrease in the rate of reaction as the enzyme is denatured. The activity of an enzyme is affected by pH. Enzymes of an optimum pH, either side of this pH value, the activity of this enzyme is decreased. The rate of an enzyme-catalyzed reaction can be found by measuring the rate at which a substrate is used up, e.g. starch by the action of amylase, or the rate at which a product is formed, e.g. oxygen by the action of catalase. Naming of enzymes, by adding suffix ase with the name of the substrate, e.g. lipids lipase, maltose maltase, sucrose sucrase, protein protease, etc. Keywords: enzymes, proteins, substrate, catalysts, condensation, hydrolysis, active sites, enzyme, substrate complex, lock and key hypothesis, coenzymes, inhibitors, seed germination, juice extraction, activation energy, optimum temperature, optimum pH. Glossary: catalysts, substances that increase the speed of a chemical reaction without themselves being used up in the reaction, e.g. enzymes. Enzymes: protein substances which act as catalysts and control the rate of chemical reactions in cells, generally speeding them up. Secretion, the production by glands of substances such as enzymes which are useful to the body. IGCSE and O-Level Smart Biology M. Rashid Chatha 030046104457. Amylase, a type of enzyme which digests carbohydrates. Short MCQs with right answers. 1. What is the substrate for enzyme X? 2. Which diagram of an enzyme, active site, and substrate is correct? 3. What is a characteristic of all catalysts? They are not changed by the reaction. 4. This sequence of diagrams shows how an enzyme works. Which labeled part is the active site? Q. 5. At which temperature does the amylase work best in human body? 37°C6. The graph shows the concentration of maltose produced when amylase digests starch at different pH values. What explains the shape of the graph between point X and point Y? The shape of the enzyme active site is changing and does not fit the substrate so well. IGCSE and O-Level Smart Biology M. Rashid Chatha 030046104457. 7. What is true of all enzymes? 8. What are enzymes made of? Protein. 9. What remains at the end of an enzyme-controlled reaction? Enzymes and products. 10. Which feature explains the specificity of an enzyme? Complementary shape of active site and substrate. 11. The graph shows the effect of substrate concentration on the rate of an enzyme-controlled reaction. The results have been obtained using a fixed concentration of enzyme at a temperature of 30 oC. Which statement explains the shape of the graph from X to Y? The enzyme active sites are not all occupied. 12. What is characteristic of all catalysts? Speed up chemical reaction. 13. What are enzymes made of? Proteins. 14. At which temperature do most enzymes, from the human body temperature, become completely denatured? 65 oC. 15. Some plants digest insects and use the nutrients for growth. What must these plants produce to digest the insects? Enzymes. 16. What happens to most enzymes above 60 oC? They are denatured. 17. The temperature of enzyme-controlled reaction is increased by 10 oC. How does this affect the rate of reaction? It may increase or decrease the rate. 18. Which statement is correct for all catalysts? They speed up chemical reaction. 19. Lactase is a human enzyme that catalyzes the breakdown of lactose in milk. At which temperature does lactase work fastest? 37 oC. 20. Which group of compounds ensures that metabolic reaction takes place effectively? Enzymes. 21. Commercial fishermen use ice to store the fish that they catch. How does the ice keep the fish fresh? Decomposer activity decreases due to decrease in enzyme activity. 22. What controls the speed of reaction of chemical reactions in all living cells? Enzymes. 23. What is true of all enzymes? They are sugars. X. They are most effective at pH 7. X. 24. The table shows the conditions in 4 test tubes containing equal amounts of starch and salivary amylase. In which test tube is the starch broken down the fastest? pH 7 and temperature 0 C. 3725. An experiment was carried out to study the effect of temperature on the time taken for protein to be digested by an enzyme. The table below shows the results. Temperature 0 C observation. 25 4 hours for completer digestion, 35 2 hours for completer. IGCSE and O-Level Smart Biology M. Rashid Chatha 030046100457. Digestion. 45 3 hours for completer digestion, 55 no digestion takes place. For these results, at which temperature does the enzyme denature? 50 0 C. 26. Which statements are correct for all enzymes? They are proteins. They speed up biochemical reactions. 27. Which statement about enzymes is essential to the lock and key hypothesis? Enzyme molecules each have a distinct shape. 28. Enzyme action can be explained by the lock and key hypothesis. Where is the active site and which acts as the lock or key? Active site, on the enzyme, lock, key, substrate acts as a key. 29. According to the lock and key hypothesis, which is the lock and which is the key for the enzyme lipase? Key, lipids, lock, lipase. 30. Protease breaks down proteins into amino acids. In the lock and key hypothesis, what is the lock and what is the key? Key, protein, lock, protease. 31. According to the lock and key hypothesis, which is the lock and which is the key? Key, lipid, lock, lipase. 32. What is the enzyme that controls a reaction in which both the enzyme and the substrate can denature at high temperatures? Protease. 33. Starch is digested by the enzyme amylase. According to the lock and key hypothesis, which is the key and which is the lock? Key, starch, lock, amylase. IGCSE and O-Level Smart Biology M. Rashid Chatha 030046100457. 6. Plant nutrition. Photosynthesis is the process by which plants make their food. They combine carbon dioxide and water to make glucose. To do this, they need energy from sunlight, which is absorbed by a green pigment, chlorophyll. Chlorophyll